The idea is very simple. We want to understand the mechanism behind cancer. And instead of looking at people who are dying from cancer, we are looking at people who are miraculously surviving after cancer. So we are looking at a very specific population. We call them the outliers because we are focusing on patients who are diagnosed at a very late stage, what we call stage 4. So when the disease, unfortunately, is metastatic. So it's not only one organ, it's 2, 3, 4 organs that are attacked by the disease, when most of the time surgery is not possible and where chemo is not really efficient. And the definition of an outlier from a clinical standpoint is a patient diagnosed in stage 4 who survive more than 5 years after the diagnosis.
Hello, Nicolas, and welcome to Impulse. It's great to have you here. I'm glad you accepted my invitation. And, as a background for our listeners, the first time that I heard about your work was during Health Europe last year. Not this year, because I think the conference took place like 2 weeks ago. I don't know if you actually went back. But it was in Amsterdam, and you were speaking on stage as part of a panel about what you are doing, studying cancer survivors or so-called outliers as a way to, understand better the mechanisms of survival and resistance in different ranges of cancers, which I found very, very interesting. So as I was thinking about, certain topics for the next season of the show, I remembered that panel discussion that you were part of. And I thought I would, reach out. So close to a year later, I think here we are, and I'm all ears and curious to understand the important work that you guys, you and your team are doing, to see a bit, what we can learn from these special individuals and how it changes from the traditional way, let's say, to understand cancer and doing research. But yeah, before we go there, and as I do on every episode of the show, I would invite you first, Nicolas, to present yourself.
Thank you very much for the invitation. I'm very happy to be here and glad that you saw me last year in Amsterdam on stage with a patient and a doctor. So my name is Nicolas Wolikow. I'm the CEO and the co-founder of Cure51, and I will detail a bit more, of course, what it is all about. I'm 53 years old, so I'm an old entrepreneur. This is my 4th company, so I already tried successfully most of the time, but I had as well some failure in the past to set up and run companies, and this is I think my biggest pleasure in life. Start fresh with brand new ideas and try to scale operations, grow the community of people working with us, and make it profitable. But getting older at some stage, what you want to do, I mean, this is personal, but that will probably resonate for other people and listeners. You want to make sure that there's a good articulation between what you want to achieve professionally and what you want to deliver as well from an ethical standpoint. And that's why, health in general and specifically oncology is a playground that I like, even if sometimes, cancer is tragic. When you can contribute in a way to improve the daily life of patients, or even better, find new treatments, new drugs that can heal cancer, cure cancer, and maybe eradicate cancer, you have a lot of satisfaction. From a business standpoint, you think you are building an asset that is valuable. And from a personal standpoint, you have the feeling that you have an impact on an issue which is unfortunately tragic and with a growing incidence in every country. Cancer, unfortunately, is growing everywhere and impacting all the segments of population. It's not only a disease that would impact all people, unfortunately. The emergence of cancer in younger generation is a global phenomenon in Europe, in the US, but in emerging countries as well. So at some stage, I thought to myself, I have to tackle this. And this is really what I'm here today.
So I saw, when I was preparing the episode, that you worked in very different environments and industries before, stepping into healthcare. So I saw that you worked at the French Ministry of Foreign Affairs, in L'Oréal, in T-Online, Dailymotion, many different areas. Yeah. And so I was wondering, how did you go into, medical technology and the field where you are now?
Originally, because a company was desperately looking for a manager in Russia, an health tech company. And for many reasons, I speak Russian fluently. So they just knock on my door and offer me the job. But I was not proactively looking for a job in health. My background at the time was essentially tech. So I work in many different tech industry, but I was very much tech driven, and what I was looking for was always something global with international exposure and capacity to work in a multinational environment. So this opportunity came. I was just got married at the time. My wife and I were very tempted to expatriate ourselves. So when the opportunity came to go to Moscow, it was 17 years ago. We said yes, and I went to Moscow. And since then, so 70 years, for the last 17 years, sorry, I've always worked in the healthcare industry. But it was originally not something that I was thinking of. So pure hazard.
So what is the idea behind, Cure51? Where does the idea come from? And, what are you guys trying to achieve?
The idea is very simple. We want to understand the mechanism behind cancer. And instead of looking at people who are dying from cancer, we are looking at people who are miraculously surviving after cancer. So we are looking at a very specific population. We call them the outliers because we are focusing on patients who are diagnosed at a very late stage, what we call stage 4. So when the disease, unfortunately, is metastatic. So it's not only one organ, it's 2, 3, 4 organs that are attacked by the disease, when most of the time surgery is not possible and where chemo is not really efficient. And the definition of an outlier from a clinical standpoint is a patient diagnosed in stage 4 who survive more than 5 years after the diagnosis. We are talking about 1 or 2% of the patients. So unfortunately, when you are diagnosed with a stage 4 cancer, most of the time, your life expectancy is 6 months, 9 months, maximum 1 year. And the oncologist will tell you, dear Nicolas, talk to your family, make sure that you can organize the rest of your life and enjoy your last moment. And still, there are these miraculous patients, these outliers who are surviving. So our job is to identify them. Very hard because it's a very rare population. In order to identify them, we set up a network of hospitals. There are more than 100 hospitals now in our network in 31 countries. And once we identify those outliers, we are collecting all their data. So it means clinical data. We have the medical file. We're going to know the lines of treatment, the surgeries, the allergies, the antecedents, all the drugs that they will have. We're going to collect imagery, all your MRI or CT scan, etc. And most importantly, we collect the tissues, the tumors, the human tissues that we are importing from 31 countries in the world. We biobank those tissues into our lab in Paris, and then we're going to do what we call multi-omic analysis. So we're going to look at every layer of the tumor cell, every layer of the genome to understand what are the biological mechanisms that explain this incredible survival. Once we decode this mechanism, what we want to do, of course, is to develop novel therapeutics, new drugs, based on this exceptional biology. So this is Cure51.
So which cancer types are you guys focusing on at first? Because I think cancer is like a general term and there are many, many different types of cancer.
Good question. We decided to tackle the most aggressive cancers, the most aggressive indications, so which are PDAC, pancreatic cancer, glioblastoma, so the brain cancer, and the small cell lung cancer, the most aggressive form of the lung cancer. So these are the 3 indications we are focusing on for now.
And as you mentioned, the description of what you call the survivors or the outliers. So you said it's people who got diagnosed at stage 4 and who survived longer than 5 years. Are there my question was, among that cohort of patients, are some kind of cured or survive fully
Absolutely.
and in remission?
Absolutely.
Or is it just a matter of delaying the...
No. You have patients with full remissions. I can give you two examples, totally opposite. If we are focusing on the pancreatic cohort, I know one patient, he had only 3 chemo, and after that, total relapse, total remission. Another one, younger, actually, when he was diagnosed in stage 4, he was 36, so very young, 134 chemos. He is surviving. It was 8 years ago. In both cases, pancreatic cancer with metastasis in kidney, so very aggressive form of evolution. And both of these patients are surviving, even though they had a totally different experience. 3 chemo versus 132. But they are fully recovered. They do sport. They enjoy their life. They have a professional activity. So
incredible. So
we have
it's
different
really
types of typologies. Yes.
So really the criteria is like, diagnosis stage 4 and more, at least 5 years of survival. Exactly. And then you accept.
Exactly. Which is exceptional. Yes. And it's a retrospective study. So sometimes, we got tissues that are 12, 15, 20 years old. So some patients, unfortunately, died, but not necessarily from cancer. Just we are retrospective. So we are dealing with a population sometimes which is very old.
Yeah. And so you mentioned, all the diversity of data and information that you guys are collecting, which I think is quite unique. What is your approach to, kind of like collecting all these data points and all these samples? Because you said you were, active in 31 countries, I think it was 100, hospitals that you are working with. So how do you orchestrate all that network?
Very good question. And in addition to the diversity of the data, we have a huge ethnic and geographic diversity. Unless most of the clinical trials, we are not only dealing with Western European or American patients. We have patients in Latin America, in Africa, in Asia. So to deal with this heterogeneity, you need two things. Ideally, you need one unique platform to collect the data. So irrespective of the origin of the data, you're going to collect the data following the same protocol, using the same tools, and curating the data the same way. So we decided to have our own platform, our own proprietary platform that we developed from scratch, as opposed to buying a software off the shelf. Due to the fact that we have this unique platform, we are sure that the data is collected the same way, with the same data point. And the other thing is that it's a clinical study. It's a non-interventional clinical study, a retrospective one, but we just have one medical protocol for the entire network. So even if you deal with a very modern American hospital or with an hospital which is, in Pakistan or in Uruguay, you are using the same protocol. So the inclusion criteria are exactly the same. The QA process is exactly the same. And all the controls that are made locally and here as well at the HQ of the company, medical control and clinical control are the same. So by having the same methodology, the same medical protocol, and the same tools, you make sure that your database is homogeneous and that you can compare one data to another.
Makes sense. My next question would be around, how do you guys approach making sense of, all that information? Because it's a diversity of modalities, in terms of, like you mentioned, biopsies or like physical samples. You mentioned information from electronic health records, additional, I guess, lab tests, clinical tests. So there's like a very big diversity of information types. So I was wondering if you can tell us a bit more about how you guys are, approaching it to make sense of all that information.
So very good question. First, in the database, for one survivor, you have what we call a control. So it's a patient from the same cohort, ideally the same hospital, and of course from the same disease, the same indication, who died, unfortunately, at the medium of the life expectancy. So most of the time after 6 months after diagnosis. And for one outlier, for one survivor, we have one control. So every outlier is matched, is paired with one control. By doing so, to answer your question, what we're going to do is to compare those two patients and isolate what is very specific to the outlier and that you don't have with the control arm. This is the first thing that we are doing. So by doing so, you're going to you're going to really enhance the differences and the specificities of the outlier in terms of biology. And then you're right, we already find a lot of signals. So the question is, are we going to find some signals, some patterns, some signatures very specific to outliers? Yes, too many. The question is, how do you make the ranking? So to make the ranking, to decide what is prevalent, what is of interest, what is significant from a scientific standpoint, you do many things. First, you look at the public literature. Some targets that we are finding have already been studied, either in academia or in some early stage clinical trials, for instance. So it's a first marker. Then you see, there's an interest. There's already scientists around the world working on that. So we probably have to keep it and work on it. And then when you are dealing with pure novelty and we are finding some new things, new patterns, new targets that don't have any equivalent in public literature, then you're going to kind of stress test them. So you're going to do a lot of tests, including experimental tests that we are doing now, for instance, on organoids. And you're going to ideally make sure that what you are finding in silico, by analyzing all this data, can be tested as well in vivo. So it's another way to refine and make your ranking more sophisticated. And then the last thing we're going to do is developing our own lab. So what we call a lab in the loop. It's a lab which is mostly driven by AI, where you're going to mix in silico and in vivo. And it's a 24/7 lab, working all time. And you're going to permanently test the robustness of those signatures and those targets. A lot of perturbation to finally get, the best of it. So the 5 or 10 targets that have no equivalent and that have probably a big therapeutic impact. And then ultimately, but we're not there yet, you go through, the typical preclinical stage, stage 1, stage 2, stage 3. But the big difference with the usual methodology is that, we already know the outcome. We know that those patients survived. So we know for sure that they have something exceptional. And B, we think we can go faster by, mixing an experimental work on a big organoid platform together with AI world or machine learning, okay? And the last thing is that we benefit from an incredible network, not only of hospitals, but of KOL, of scientists. We are working with, the best oncologists in the world. So each time we isolate something we think it's worth of interest, we're going to share it with the community and tell them, this target in glioblastoma, these are the characteristics. What do you think, guys? Should we continue? Should we deprioritize? So it's really a mix between AI and machine learning and algorithm. But of course, as well, we capitalize a lot on the knowledge and the expertise of our physicians all over the world.
Understood. And so going back to the control arm and the control individual for each survivor in the cohort, they are also matched in terms of the treatments they obtain, right?
Absolutely. So, yeah, the lines of treatment they have. Absolutely. It's part of the criteria. Exactly. So we're going to match together, for instance, if you got FOLFIRINOX in first line for PDAC, you're going to be matched with a control we receive the same treatment. Absolutely.
Understood. What are some of the, early signals, I would say, from your research? Are there already certain findings you can share at this stage?
There are some early stage findings, especially on the PDAC cohort, because this is the one that we prioritized. So this is where we put maximum effort in terms of science and bioinformatician team efforts. So we find already just after 3 months, we started the sequencing, not the collection, but the sequencing just 3 months ago. We already find 2 targets, novel targets, which have been already validated experimentally. So what we did in 3 months sometimes, takes 4, 5, 10 years in the traditional industry. So it's very encouraging. Again, it's very premature. There's plenty of things to do to validate those assumptions, but it's very encouraging.
And have there been any surprising, let's say, biological or genetic findings from the cohorts so far, like something that, maybe
Again,
for one individual?
We need to have a new conversation in 6 months or 1 year. But now what we find sometimes, and if I keep talking about the PDAC cohort, we find some expression of genes that are usually associated to other diseases. Other indications, like colorectal, for instance. And we find them as well in the cohort of the PDAC survivors. So maybe there's probably kind of a repurposing things behind it. And the overexpression of a gene, which is traditionally not associated to PDAC, but to something else in the PDAC cohort, might explain partially the survival. So it's very interesting for the next step of the analysis. We might have, some crossroads at the intersection of different cohorts. So maybe the clue to the survival of, small cell lung cancer patients will be found in glioblastoma or in ovarian or in stomach. So it's the interactions between the tumor cells and its microenvironment is not necessarily always attached to one specific disease. So we can have some pan-cancer explanations. So it's very interesting as well. This is something we are looking at.
When I was, thinking a bit about your approach, I had the following question that came to my mind. So I was thinking, like, what is the level of evidence that we have, that kind of like indicates that what we would learn from the biology of these survivors can be kind of like replicated or applied to, other patients who might not have those properties? Because I'm thinking, like, if it's a certain, genetic expression profile, or if it's other aspects of what makes these persons unique, how can we, translate this into, I don't know.
It's a very good question, and I'm not sure I have. Yeah, it's a very good question, and I'm not sure I have all the answers. There's something I must say that I forgot to mention in the introduction. I'm not a PhD. I'm not a doctor. I'm not an oncologist. So I'm surrounded by experts much better than me in those matters. So I'm going to give you first answer, but at some stage I have to stop because I'm not legitimate and I don't have the skills. But you were asking me, what did you find so far? What we are finding so far, and again, this is really the beginning of the journey, is that most of the things that we are finding are exceptional response to treatment. Okay, so this is something that those outliers have in terms of gene expression that makes the treatment extremely efficient. Remember, this patient, after 3 chemo, he was fully cured. So this is what we are finding so far, as opposed to immune system or exceptional biology. Maybe if we have another conversation in 3 months, I will tell you something else. But so far, this is what we can find. So if this is one of the clues, then it's easier to adapt it to the rest of the patient. You just have to develop drugs with that will overactivate that gene. But again, very premature.
Yeah, understood. So we'll need to have like another.
Yeah, and that's why as well. Sorry. Yeah, the only thing I can add, this is what we mentioned before. The fact that every outlier is paired to a control is really to make sure that we can find commonalities with these two populations. We are looking at differences, but we are looking as well at commonalities. It's very important. We are looking at both, actually.
Understood. So, you've spoken, I think in your introduction about, and I think in some other interviews about eradicating, cancer from Earth, which is a good mission, I guess. I wanted to ask you, what does success look like for you in, maybe in the next 5 years, like let's say midterm?
So we never said in 5 years. We said in 15 years or in 10, from 10 to 15 years. But as you said in your introduction, unfortunately, cancer is very complex and it's a pluri-indication disease. So I'm not sure we're going to find one clue to kill all cancers, but that's why we decided to start with the most aggressive ones. And these 3 cancers are not only the most aggressive ones, these are the ones where the therapeutic landscape did not evolve at all over the last 30 years. PDAC is a good example, but this is the same for glioblastoma. It's a tragedy. The worldwide community, the scientific community doesn't find efficient drugs for glioblastoma. So the first satisfaction for us, the first major achievement will be to find what we call targets. So therapeutic target, validated, robust for each of these 3 indications, that we can then develop in order to develop new drugs. And the second thing, we want to go faster than the usual industry cycles. On average, to develop a drug, till FDA approval, it takes from 9 to 12 years on average, sometimes 20 years. We think that due to the fact that this data set is exceptional and unique, and mixing up scientific expertise together with AI, we think we can reduce those cycles by 2 or 3. So to answer your question, I would be very proud when we would put on the market our first drugs, and hopefully in less than 7 years.
Yeah, that's an ambitious goal. I was also thinking, when I was looking at your details about your study and your general approach, beyond oncology, do you think this survivor-centric and data-driven model can also be applied to some other disease areas?
Probably. Of course, some neuro disease, when there's a survival population, Parkinson might be a good example, for instance. When there is a survival population, we might probably adapt the same methodology, identifying the survivors, collecting the data, having a control arm, and see how it looks. But again, I'm very cautious because as opposed to the outliers in oncology, where we have a lot of articles and a lot of works that have been done by the academia, I don't know if there's the equivalent for neuro, for instance. There is probably, but we never work on it. But that should be possible. And again, the beauty of this network of hospitals is that most of the time we are dealing with the oncology part of the hospital, but when we are dealing with big hospitals, they have as well a neuro unit. They will have a rare disease unit as well. So that once you have an established relationship with the hospital, the data collection, when you go to other therapeutic areas, will go faster. Your contracts are already established. Most of the rules in terms of budget, privacy, data management, transfer are done. So you can accelerate the data collection because 80% of the paperwork are already done.
Yeah. That makes sense. I wanted also to understand a bit better, like maybe from it's more like a commercial or a business perspective, like how you guys are set up. How is, the current research funded? How are you going to get, returns on that down the line?
So 2 answers. First, we are privately funded, totally privately funded. We made what we call a seed round. A first fundraising last year for 15 million euros as a seed fund with 3 investors, one in Europe, one in the US, and one in Japan. So it was important for us to have this international footprint from day one. And we are working now on a Series A, which will be much more significant in order to grow the platform, launch new indications, new cohorts, and develop our own lab. And in terms of business model, to tackle the second part of your question, the business model of the company is very simple. I was talking about our capacity to isolate targets. The idea is to have a catalog, a library of targets, and you can have 5 or 10 targets per indications. And in 4 years, we're going to have 12 indications. And each time you have a validated target, you choose the best partner to develop the drug. It might be a pharma or a biotech. We keep the IP because, notable fact, Cure51 owns 100% of the dataset. So the data is our property. So we own the dataset, and you have a deal, a traditional pharma deal with milestone and royalties. And you have a revenue sharing agreement with the pharma. But again, the beauty of the model is that you can duplicate it many times for each indication. So this is the business model of Cure51. Developing, finding new targets that are validated, and then finding the best partner to develop the drug. Our business is not, we are not a biotech, we are not a pharma company, we are a tech bio company. So we have a platform, we have a network of hospitals, we find patients, we collect their data, and we understand the mechanism beyond the survival.
Very clear. How did you convince or you mentioned IP and you mentioned that you're, the sole proprietary of the data set. How did this work with the different health systems? Because, we recorded an episode, I think it was episode 40 with Jean-Philippe Vert from Okin that you probably know. And he was explaining us their approach, which they use like federated learning to leverage local databases. And I guess they don't own the full data set. So I'm curious to hear.
We convinced the hospital to work that way with us around, I would say, 3 pillars. The first one is that we have a revenue sharing agreement with all the hospitals. 10% of the future revenues of the company are shared with the hospitals, depending on the number of patients they brought to the study. Second thing, when they give us a cohort, let's say, of 10 patients, I would make the full molecular sequencing of those patients, and I will give them back the data. So each hospital has access to the raw data of its cohort. So by doing so, you contribute to the scientific effort. It's important for hospitals in France or UK or in the US. Imagine for a hospital in Uruguay or Africa, where they don't have access to those kind of technology. It's incredibly precious. And the third thing is that we are creating a scientific community where we make sure that any oncologist from our network can talk to his peers. We make sure that, and very soon you will see it, we have co-publications, and we try to encourage, and accelerate research work inside the network. So we are creating a scientific community as well with a very renowned oncologist. And I think it's another factor that can explain the motivation of the hospitals. And maybe the last thing is more cultural. We are a startup. We are not a pharma. We are not a corporate. And we are not backed by any pharma, actually. We don't have Sanofi or BMS in our capital. And I think the fact that we are totally agnostic, it's something that hospitals appreciate. We are independent and agnostic. And we try to go fast. And they like it, actually.
Yeah. No, thanks for sharing. I think it's also quite unique. I mean, that model seems to be also quite unique. I guess you're always open to have more cases to the database. Like, is there a certain number of,
There is according to the medical protocol, this is the magic number that the scientists gave us. To have enough statistic power, you need 132 outliers per cohort. So it means 132 outliers and 132 control. We decided to increase that threshold to 170, just to have kind of a contingency. So 170 outliers, 170 control. But actually, we don't put any maximum limit. So we keep collecting the data. I mean, the outlier, finding one outlier and have access to his data is so precious that every time we have access to a patient that meets the inclusion criteria, we take it into the database.
For if there is someone from a health system or oncology unit listening to us and who would like to maybe submit a case or get in touch with you, how are you kind of like recruiting or establishing new collaborations with health systems that are not part of the network yet?
You mean how are we onboarding new centers?
Yeah, exactly.
Yeah. Most of the time, I mean, not most of the time, actually, all the time. We are starting with PIs. So we would identify in a given hospital one PIs or two PIs, working on the indications from our study. Then we will engage to those PIs. We will share the medical protocol. We will explain the approach, and then if we think that they are motivated and they want to participate, they sign an NDA, we are sharing the doc, we negotiate a contract. As we are the sponsor of the study, we would pay for all the operational expenditures and costs. So number of days that the PI will dedicate to the study, the same for the CRA, the same for the biobanking cost, etc. We negotiate a data management transfer agreement, and then we start, collecting the data. So most of the time we are approaching the centers. Sometimes centers and hospitals come spontaneously to us, and most of the time through our website. There's a section in the website where you can apply or when you can ask for more information, and this is how we establish the contact.
Mm-hmm. Makes sense. I'll put the link in the show notes to I think it's called the Rosalind study, right?
Yeah, the Rosalind study. It's a tribute to Rosalind Franklin, the famous British oncologist. And this is why, as well, we are called Cure51. It's a reference to the cliché number 51, when she, for the first time, identified the structure of DNA.
Interesting. Yeah, I had not made the link. Okay.
Yeah.
Cool. No, it's very interesting. I mean, I may I ask you, like, until when, or according to the protocol, like, until by when should the study be finished?
Never, because we're gonna increase the number of indications every year. But for these 3 first indications, we think that we're gonna end up the collection in December, so at the end of the year. By December, we would have probably more than 700 survivors and 700 controls for these 3 first indications. And then next year, we're going to launch 2 indications, and the year after as well.
Makes sense. Cool. No, I'm really, impressed. I'm looking forward to seeing, what comes out of.
Me too. Me too. We are all excited.
I thought it was quite interesting that, I mean, I'd never heard that approach of like looking into survivors to understand conditions in other disease areas, which in a way is a bit surprising because it would also be a logical, way to think about it. But I didn't make a search if there are like, other, let's say, initiatives.
There is another initiative that inspired us a lot, actually, but it's not in oncology. It's a US company called Variant.bio, and they have the same approach, so retrospective approach, but they will look at what they call the superhero. So people, who have incredible physical capabilities. They just sleep 2 hours a night, or they have some blood pressure, which is incredibly low. So this is the population they are looking at. So it's a bit different than us, but the approach is the same. And this is a company we looked a lot with my co-founder, and it was an inspiration.
So more on the longevity.
Kind of, exactly. Exactly. Not in oncology, more in longevity, absolutely.
Makes sense. So, I'm conscious of your time, Nicolas, and I'm very thankful for, you coming to the show and taking us through your story. For our listeners, who might be interested in learning more about your work and, let's say, by extension, having, being interested in the field in which you work, like, would you have specific resources you would like to redirect them to? Could be books, publications, websites. We
did not publish yet, unfortunately, but we will soon. I hope that for the next ESMO, we will be able to publish something. But but they can contact us through the website, and especially if it's a physician or oncologist, we can share our white paper, we can share the poster of the study, we can share the synopsis of the medical study, we can share the list of the hospitals we are working with. So there's I would appreciate any spontaneous application for people that want more information about the project. We are very transparent. It's one of our key value, transparency. We're very transparent with the hospitals. We're very transparent with the staff, and we are very transparent as well with the medical community in general.
Yeah. I mean, let's include as well those details in the show notes, then people Absolutely. Can find them directly.
Okay.
Could you share with us, maybe it's again a bit early to ask the question, but as I ask it all the time, I'll still do it. Can you share with us an anecdote from your work at Cure51 that already kind of like made you realize the impact that you were having on people's lives?
I can tell you the first patient we talked to, because again, the beauty of this project is that, yes, it's a database. Yes, we are talking about a tech platform, but we are exposed as well to patients every day. So it was really early stage 3 years ago. At the time, we had nothing. We didn't have a product. We were starting, having conversations with hospitals, trying to convince hospitals to work with us. We were auto-financing the project. It was before the seed round. And I was, surfing on Facebook, on Instagram, trying to find communities of cancer survivors. And I found a patient in the US, in Florida, Camille, who survived 12 years to pancreatic cancer. And I just sent a direct message, thinking to myself, there's no chance she answers. I'm not an oncologist. Nobody knows me. She answered immediately. We started engaging, and she's the one, actually, who found the signature of the company, Hope is for Everyone. Cure51, the tagline is Hope is for Everyone. This is coming from Camille. And since then, she was our first ambassador. She's a member of our patient committee, and she's really active, in advocating for the cause. She's helping us to be more visible in the US. But the fact that she trusted us from day one, even though, again, it was only two crazy French entrepreneurs with this crazy idea, with no medical background, was, a very strong signal for us, which is still resonating today, saying she did survive. She wants to share her experience. She wants to give hope to every patient who unfortunately is suffering from cancer. We need to make sure that this dream, this ambition can be achieved. And this is a source of motivation on a daily basis for us.
Thanks for sharing that. If you would recommend a fellow healthcare innovator or as a potential guest for the show, who would you recommend?
These people from Variant.bio. They already negotiated, first deal with the pharma industry, so they were able to value their incredible superhero database. They are based out of Seattle. Very nice company. I know some people there. One of their co-founders with one of our advisors, actually, and it's an incredible company. They work a lot with African population in Kenya and Ethiopia in particular, if I remember correctly. So it's a kind of a fascinating company as well.
Variant.bio. Okay, well noted. I'll check them out. Nicolas, thank you so much for taking us through what you guys are doing. I think there's probably a lot of curiosity that was created throughout the conversation. So I'll invite also our listeners to follow the rest of your adventure. I guess you're present on different. How can we actually get in touch?
Instagram, LinkedIn, Facebook. We present on the three networks.
Super. Yeah. Then I'll also put all the links in the show notes. Thank you so much.
Thank you very much. Thank you. Merci beaucoup, Mathieu. Thanks.
Thanks for watching the episode. I hope you enjoyed it. You can subscribe by clicking on the podcast channel at the top right and watch another episode here at the bottom.